Now, tongue rolling is a dominant trait. So, there would be tongue rollers and non-rollers. So, this is how the tongue on rolling appears. The people who can roll their tongue, the lateral side, they can be folded inward, and that's what is tongue rolling. So, suppose in a population, the non-rollers, that means who cannot roll their tongue, non-rollers are 16 out of 100. So in this case, we have just mentioned that rolling of tongue is a dominant trait and non-rollers must be Q square, that is having the recessive trait, and if we convert it to decimal, it would be this, and that means Q would be 0.4. Now with this, we can get from the first equation P, because we have seen that P plus Q is 1. So that means P is 1 minus 0.4, that would be 0.6. So now from this we can calculate homozygous rollers and heterozygous rollers. Homozygous rollers, for this we require P square, and P square we have just now calculated the value of P, and that is 0.6 whole squared. This will give us 0.36. And heterozygous rollers, this would be 2PQ, and putting the values, that is 0.6, Q is 0.4 into 2, so this will give us 0.48. So with this simple equation, we can find out the frequency of various genotypes in the population. That means homozygous rollers are 36% in the population, heterozygous rollers are 48% in the population, and non-rollers are 16%. And if the population is in equilibrium, generation after generation, the frequency is going to remain the same. But if there is any genetic force operating, it is going to disrupt this equilibrium. So what are the various factors which can disrupt the equilibrium? And those are, of course, mutations, gene migration or simply migration, another is gene recombination, natural selection, and last but not the least, genetic drift. So we will consider some of these factors, out of which some we have already considered along with synthetic theory, like mutations, gene migration, gene recombination we have already mentioned. Selection and genetic drift need to be discussed further. Now before we delve into that, let us first solve a few questions based on Hardy-Weinberg equilibrium. So here we have a question: at a particular locus, frequency of A, that is dominant allele, is 0.6. That means here we have been given P. P is 0.6, and that of recessive, that is small a, this is given as 0.4. So what would be the frequency of heterozygotes in a randomly mating population at equilibrium? Now for heterozygotes, we have seen that it should be 2pq, we'll have to calculate. So this is 2 multiplied by 0.6 and multiplied by 0.4, which would be 0.48. So here in the option number 4, we can see it is given, and that would be our correct answer. Now let's take up another question. In a population of 1000 individuals, 360 belong to the genotype capital A, that means homozygous dominant, 480 capital A small a, that is heterozygotes, and the remaining 162 homozygous recessive. Based on this data, the frequency of allele A in the population, capital A, that is dominant. So allele A, that means we have to find P. Now in this case, the question seems very intricate, but actually it is very simple. And here we have seen that capital A, which would be representing what? P square. This is given to us as 360. 360 out of 1000. So we'll just take up P square is this, or we can convert it into the decimal form. So here P would be under root 0.36, and that is 0.6, right? And here the option that we can see here is the third one, and this would be our correct answer. So rest of the data was not required. Just by one single data we can easily calculate in this question. Let us now discuss the various genetic forces which disrupt the Hardy-Weinberg equilibrium, in which we will take up selection and genetic drift. Now moving on with the factors which disrupt the Hardy-Weinberg equilibrium, in that again coming on to natural selection. Now natural selection we have already studied along with the synthetic theory, but now we are going to take up the different types of selection. Now one of the type is stabilizing selection, which is the most common type of selection occurring in nature. And this operates when the environmental conditions are constant or they are not changing, and in which the selection favors the intermediates against the two extremes. Now let us first see here a particular graph in which we are taking one kind of trait, let us say the height and the number of individuals who are of this particular trait. Now for any trait, if we will plot a frequency polygon, what we will find is that there are some individuals who are showing one extreme. That is, even in a crop field if we see, there would be some plants which are very short in height, while there are some plants which are very tall, but maximum plants are of intermediate height. So that's why we get this kind of frequency polygon, where the two ends are showing the extremes while the middle region is called the intermediates. And this line here is the mean value. So when we plot this graph and we get like this, but what is selection doing? Selection is actually favoring the intermediates. And when we say favoring, that means they have a higher fitness value, they will be adding more offspring to the population, and over a period of time, the number of intermediates will increase in comparison to the two extremes. And this is how the graph will now appear. So here we can see that now there are more number of individuals near the mean value while the extremes have decreased. So their representation in the population has decreased. And this kind of example is even seen in the human population, like birth weight of most human babies is usually in the range of 3 to 4 kg. And it is seen that much smaller or much larger weight babies, they suffer higher rates of mortality. So it is the intermediates who are being selected. Now moving on to next type of selection, that is directional selection. Now, directional selection would be when the environment is undergoing a change in one particular direction. That means it is continuously becoming colder or drier, something like that. That would be when directional selection would operate. And in this case, what is going to happen is that one of the extremes would get selected. So here, this is our original frequency polygon, and out of that, any of the extreme, whether on the left side or on the right side, may get selected. And as a result, what happens? That this entire graph will be shifting to one side. And now there would be a new mean. So the population will be acquiring the character which is other than the mean value. So it is some other value but not the mean value. And one of the best example in favor of directional selection is industrial melanism, where we had seen that with the increase of population, the dark moths were being selected. And even here we can give the example of giraffes. Like in case of Lamarckism, he had explained that giraffes got long necks because of stretching. But according to Darwin's natural selection, it can be explained that all types of giraffes were present. That means there were short-necked, there were intermediate-sized necked, and there were long-necked giraffes. And when there was drought for number of years, then only the long-necked giraffes could forage, and they were the ones who survived. And ultimately, that led to the development of a particular evolutionary lineage, that is only the long-necked giraffes are now present. And all artificial selection, because humans, they select for the best yield out of the plants or the animals, and that is selection of one extreme, and that is also directional selection. Now the third type of selection, that is disruptive selection, which is a rare type of extremes get selected. And this would be when the environmental conditions are not homogeneous, they are continuously fluctuating. And ultimately, what is going to happen? It may lead to the evolution of two different species. Now again, let us begin with our original frequency polygon, which we have seen, and here of one extreme, and that is also directional selection. Now the third type of selection, that is disruptive selection, which is a rare type of selection where both the extremes get selected. And this would be when the environmental conditions are not homogeneous, they are continuously fluctuating. And ultimately, what is going to happen? It may lead to the evolution of two different species. Now again, let us begin with our original frequency polygon, which we have seen, and here both the extremes, if they are selected. So over a period of time, it will be like this. There will be two peaks. That means the population will acquire values on both sides of the frequency polygon, and those are the peripheral values. And here we have species resembles a unpalatable species. So the females, they have two models. One is of course a distasteful species, and the other if they are selected. So over a period of time, it will be like this. There will be two peaks. That means the population will acquire values on both sides of the frequency polygon, and those are the peripheral values. And here we have some rare examples, but one of them is Papilio dardanus. It's a butterfly found in Africa. And in this case, the females show Batesian mimicry. And remember, Batesian mimicry is where a palatable species resembles a unpalatable species. So the females, they have two models. One is of course a distasteful species, and the others, they resemble the males. They are endomorphs. And any of the females who are intermediates, that means they are not completely mimicking any of the model, then those are the ones which are easily recognized by their predators, and they are becoming lesser. And we can imagine over a period of time, these intermediates may become lesser and lesser, and these Now these three selections, they were based on phenotypes, and anyway we have earlier also discussed that natural selection acts on phenotypes. But there is one rare type of selection that acts on genotype, and that is balancing selection, or which is also called balanced polymorphism. Now if a gene has two alleles, which usually are there, and out of those two alleles both are maintained in the population, so that could be only by maintaining heterozygotes. So heterozygotes have an advantage over homozygotes, and this way both the alleles have the adaptive value, both the alleles are getting selected. And one of the best examples we have from here is sickle cell anemia and G6PD deficiency. Now sickle cell anemia, of course, is a genetic disease in which the beta chain of hemoglobin is defective. It is just a point mutation where glutamic acid has been replaced by valine. And G6PD deficiency is a disorder in which this enzyme, that is glucose 6-phosphate dehydrogenase, this is not formed. As a result, RBCs are fragile and they easily rupture. However, both these diseases are found in the areas where also malaria is an epidemic. So that shows that here a deleterious gene in the heterozygous condition is providing an advantage to the individuals because they are resistant to malaria. And during epidemics, the homozygotes who are for normal hemoglobin or normal RBCs, they would be dying. The people, those who are homozygous for sickle cell anemia, they are highly anemic and they don't survive beyond childhood. And even here there will be massive hemolysis, and that's why they don't survive. But the heterozygotes have an advantage. So this way even a deleterious gene provides some advantage, but in the heterozygous condition. In the homozygous condition, of course, it is highly harmful for the person who is having it. So this was regarding the selection. Now moving on to another factor which disrupts the equilibrium, and that is genetic drift. Now genetic drift is something which is totally opposite to selection, because selection is a filter which is filtering the alleles which are providing some advantage to the individual. But genetic drift is totally a random process. It can select anything, whether that allele is adaptive or not. Maybe it is non-adaptive, maybe it is neutral, that will get selected. So it is basically a statistical accident. That is, in statistics, we take out just a small sample and that sample may not be the best one. And this can occur when a population undergoes certain kind of random events where many individuals, either they are killed or they move from one location. As a result, some of the alleles may be totally lost. So the individual alleles of a given gene are all represented in few individuals now in the population, and some of them may be lost from the population altogether. And this genetic drift can operate in two ways, which are founder effect and the bottleneck effect. So let us take each one of them one by one. First of all, the founder effect. Now founder effect is when few individuals, they move from one location and colonize another region. So here, what is the situation we can see over here? Let us say this is the initial population. Now out of this initial population, few members, they migrate out. Let us say these are the founders. And in these founders, you can see there is one combination. This is also going along. And now when they make a new population, so we can see that this small a, the mutant allele, its representation in the new population has increased. This is what is called founder effect, because the alleles brought by the founders, that is enhanced in the new population. Now the founder effect is particularly important in evolution of new species on islands such as Galapagos Islands, and there are greater chances of even rare genetic diseases among island populations. So here it led to speciation on Galapagos, but usually in the human populations on islands, if we see, there is more representation of rare type of genetic diseases, which shows that some heterozygotes had come with the early colonizers, and because of inbreeding, the representation has increased. Another is genetic bottleneck. Now, genetic bottleneck is quite similar, but here the original population undergoes a population crash. That means many of the individuals are killed and only a small group of survivors is left out. So some of the alleles are totally lost, and loss of alleles means loss of variations. And such a population, after inbreeding, it will develop into a weaker population, like it is seen in the cheetah population of Africa. Now these African cheetahs, they were hunted upon, and as a result their population size decreased. Now they are inbred among each other. The Asiatic cheetah are already extinct, so there is no way out that they can be brought back into a situation where they are free from the danger of extinction, because all those rare genetic alleles which makes a individual prone to number of diseases, they are expressing themselves and they are likely to become extinct. So this bottleneck effect again we can show here with a diagram. Let us say that these are the various alleles which are there in the original population. Now this population undergoes a crash and only few alleles are left. So the frequency has undergone a change. Now whichever are surviving, which are there here in this bowl, they will survive into and they will form a new population. And over a period of time, what we see that in the initial population we can see two different colors, that is just to represent two alleles, but now one of the allele is more represented in comparison to the other one. So this new population has a new combination which may be making this population very weak. And in many of the animals it is seen that this condition cannot reverse the path of their extinction. Now, one last factor, that is non-random mating. We had said that Hardy-Weinberg equilibrium operates if there is random mating. So non-random would be opposite, that is when individuals who are sharing their genotypes, among them there are matings, like inbreeding. Mating with the relatives is the most common form of non-random mating, and as a result what would happen? It does not alter the allele frequencies, but it decreases the proportion of heterozygotes. 2pq would decrease and p square and q square will increase. So certainly the equilibrium has been altered. Now let us take questions. Frequency of an allele in an isolated population. So small isolated population, that is our keyword for genetic drift. So here the answer is first one. Next, sickle cell anemia is characterized by elongated sickle-like RBCs with a nucleus, an autosomal linked dominant trait caused by substitution of valine by glutamic acid. This is opposite, and caused by change in a single base pair of DNA. So this is our correct answer. That is option 4. And next one, artificial selection to obtain cows yielding higher milk output. Just now we mentioned this would be directional selection. So here in the option 2 we can see that it is directional selection mentioned. So this is answer 2. And G6P dehydrogenase deficiency causes the hemolysis of, so here hemolysis of RBCs, and it is the 4th one. So this brings us to the end of today's class, in which we have discussed synthetic theory and Hardy-Weinberg equilibrium. Next we would be taking up speciation and of course our own evolution, that is human evolution. That's all for now. Have a nice day.